Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Cut and Paste of Myosin Binding Protein C in Striated Muscles, featuring Dr. Samantha Harris, Professor of Physiology at the University of Arizona, and Matthew Borkowski, General Manager at Aurora Scientific. Today, Dr. Samantha Harris will discuss the development of 3 new mouse models in her lab, engineered to target and replace specific myosin-binding protein C paralogs in muscle fibers and impact of their mutations on skeletal and cardiac muscle diseases. We'll also hear Matthew provide some context into the Aurora Scientific myofibril system, specifically designed to measure the nanonewton forces arising from activation of single myofibrils. I also want to take this moment to thank our partners at the American Physiological Society and the sponsor for this event, Aurora Scientific, for making it all possible. And so with that, I am ready to welcome our first speaker, Matt, to join me.
Thank you, Sydney.
Thanks, Matt. Take it away.
I will. All right. Hello. Welcome, everyone. Thank you for attending our webinar here about the tools and techniques to study the contractile properties of myofibrils. Today, I will try to give you an overview of the equipment and the techniques themselves. And later on, my co-presenter will describe a little bit about her current research and touch a little bit on how myofibril contractility fits into that. Before doing that, though, I'd like to take a moment to just briefly tell you a little bit about Aurora Scientific. So for those of you that have attended one of our webinars before, no doubt you've seen this slide from me. Aurora has been serving the muscle mechanics community since 1997, when we began selling only R-force transducers and positioning motors. Since then, we have become a trusted partner of the community by developing stimulators, software, and experimental apparatus to package our instruments into turnkey systems. And these turnkey systems range from testing forces produced by whole large animals like dogs and pigs and sheep in vivo, all the way down to testing isolated single skin cells and fibers on the small end. It's that small end here that's how it used to go. And today we'll be looking at our integrated system for single myofibrils, which represents the smallest contractile unit above the actual actin and myosin proteins, and the smallest sample, which we have the ability to assess currently. So what can I say about our single myofibril system? Well, although it is technically the newest complete system in our catalog, its development journey began more than a decade ago in partnership with our close colleagues at McGill University in the laboratory of Dr. Dilson Razier. During that time, a really brilliant optical design to measure AFM deflection was designed and patented in the lab by a physics PhD student there named Alex Labuda. That design would be colloquially referred to as the penguin, as the optical periscope resembled a penguin's beak. This penguin, if you will, is an AFM transducer that would essentially form the foundation of a low compliance and powerful test system to measure these contractile forces in single isolated myofibrils. This was not the first system to do so, though. Novel methods of measuring myofibril force were described in the literature in the 70s and 80s. And the bending cantilever design, another common technique, was in use considerably earlier than this penguin system. However, thanks to the close collaboration between McGill and Aurora Scientific, we've taken this AFM-based system and refined it into our 1700A. A system that is repeatable, reliable, and offers a powerful means of accurately quantifying this sub-micronewton level forces, tens of nanonewtons typically. So all this history is very well and good, and while this development journey is certainly an interesting one, we're all here to hear a little bit about more of the nuts and bolts of this system. So how does it work? So let's start with this AFM benting cantilever design, which we call our 470A AFM transducer. A simple schematic can be seen on the diagram here in this slide. And essentially, the myofibril is attached, typically with a silicone-based adhesive, between a very stiff microneedle and the probe of the AFM cantilever itself. From the head shown in the previous slides, a conjugated laser beam is focused on the AFM cantilever by positioning the cantilever as near as possible to the laser's focal point. Any deflections of said cantilever, which has a polished gold surface, will then refract some of the conjugated beam back up through the optical periscope onto a 4-quadrant photodetector cell. The intensity and the position of the reflected light hitting the photodetector represents a change in deflection of the AFM and thus a change in the contractile force produced by the myofibril. The stiff needle is typically coupled to a fast piezo, which can be used to induce stretches or slacks of the myofibril prep, which is deflecting what the AFM quantifies as force. So just a few more words about the cantilever itself. The stiffness, the nominal stiffness of the supplied cantilevers are about 0.2 newtons per meter, which is actually relatively stiff given the small forces involved. Typical myofibrils will produce a deflection on the most of a micron or two. So this is still relatively low compared to the total preparation length. However, we know that the actual stiffness of the cantilever can vary quite a lot compared to the nominal quoted stiffness and what the true cantilever stiffness is. So thus, we must perform a calibration. This is achieved by physically moving the cantilever with a known displacement, which in turn is achieved by this piezo activator I just mentioned. So a few words about the piezo. It's our model 340A, which we use as this very rapid actuator to alter the length of the myofibril preparation. The design is based on a deflecting or bending piezo stack, which deflects or bends the tip of a stiff glass needle. This is typically fabricated with a pipette puller. The piezo motor is designed to work with standard 1 millimeter glass pipettes, but there are other sizes available upon request. The exact amount of movement that the piezo can achieve is dependent on the precise length of that stiff glass needle, but this is typically on the order of about 50 microns of total movement. So the shorter the stiff needle, the less compliance that will be expected at the piezo end of the connection, but even with these sort of typical values I've just cited, we would expect to see sub-micron compliance on this side. So I've foreshadowed a little bit describing the two preceding instruments about the importance of calibration. And with this system, a good calibration really depends on precise alignment of all these instruments and the laser in a 3D space. So firstly, one, we must ensure that the distance from that optical periscope to the mirrored face of the cantilever is set such that it is close as possible to the laser's focal point. Since the focused laser spot is still going to be considerably larger, its diameter that is, than the width of the AFM itself, this will ensure that that refracted signal is maximized as much as possible. Secondly, number two, we must adjust the lateral position of the cantilever as well as its height relative to that spot, the focal spot of the beam, such that we make sure it's focused on the bending portion of the cantilever and not the large mirrored area at the top. And the best way to do this is to find the bottom edge and then move from side to side and move up and down to maximize the signal. We also want to know exactly how far we are up that bending portion of the beam. Thirdly, and finally, we must adjust the position out of the laser's periscope to find the maximum signal response profile across that entire range, as you can see on this representation of the quadrant detector. So just from this simple diagram, we can sort of see that calibration is a multi-step process of precise alignment. And what tools do we have to help us with these tasks. So some of you may already be familiar with our 600A digital controller. This is our real-time data acquisition software that we've written. The software acts as the master timer to control and record all aspects of the experiment with sub-millisecond precision. However, we've now built in all sorts of applets to facilitate the capture of the diode and nose profiles, background profiles, as well as controlling perfusion and measuring the force of the bending cantilever during calibration. The data files are all still stored in this same open source format as before. And calibration applets and subroutines and wizards in the software are all well and good, but it doesn't necessarily help us align the AFM and the laser and all those other associated pieces of the system that you've sort of seen from the photos in the previous slides. So for this purpose, we introduce our purpose-built 820A motion controller. And this is a hardware device that encodes the position from the motorized micropositioners so that in this navigation tool that you can see here, we can visualize and remember the position of the AFM and the stiff needle in 3D space, which is really powerful. Once we've remembered our ideal position relative to where that laser spot is, we can return the instruments there automatically with the push of a button. And that positioning accuracy is much less than a micron. So that's a real powerful way to not have to sort of find this alignment over and over and over again each time. Finally, we have a high-speed camera for sarcomere spacing detection, which is really incredibly useful as a means of measuring also precise spacing between objects. So in this case, we're able to accurately set the AFM near the focal point, near some of where we expect the laser to be at its highest focus, since this magnitude is approximately known. So we're already starting, in a reasonable place. Whereas the preceding 820 positioning tool was useful for remembering precise locations, the camera will very precisely measure what we see in the eyepieces of the microscope while trying to get the alignment of the tools or to validate the magnitude of the bending of the AFM from the piezo motor, as that piezo is open loop. Expansion modules for edge detection are presently under test and development, which is actually sort of interesting for those of you that are initiated to these techniques, because there is another commonly employed method for making contractile measurements, which I think is worth discussing. As shown on the left, the leftmost image, that is, it involves two stiff needles, which, as you can see from the photo here, are of different sizes and thus of different stiffnesses. The stiff needle is meant to say essentially stationary, or it can be attached to a simple actuator, and the deflection of that thinner bending needle is observed with a high-speed camera directly. So as you can see from the two pictures side by side, the bending glass needle system is clearly simpler than our AFM-based system. So why bother to use ours? Why would anyone concern themselves with it? And so you can see from this little comparison table, ultimately it boils down to compliance, at least as a leading reason. The much lower compliance of the 1700A AFM offers this ability to do a wider range of dynamic experiments that involve changing length of the myofibril, particularly very fast changes. The bending needle system is considerably simpler and makes positioning of the tooling easier, and both needles can ultimately be fire polished for glueless attachment of myofibrils, which is a big advantage. But in terms of calibration, while the bending needle may be simpler at first glance, there are assumptions made about attaching each myofibril in the same spot relative to the length of the cantilever. And it still requires a sensitive transducer, as quite often our own 406 is used to obtain the stiffness of these bending needles. no system is perfect, but considering the right approach for making these measurements is extremely important. So, in summary, the low compliance of our AFM makes it an excellent choice for a true isometric measure of myofibril force, and it's an excellent choice for any protocols involving dynamic changes of length, particularly fast ones like KTR. The replaceable nature of the AFM cantilevers mean that different stiffnesses can potentially be dropped into the same system to study different preparations, and that there is always a constant, reliable source for parts, support, and service. So with that said, many thanks for joining for this brief overview presentation and for your interest. Thank you.
I just wanted to start by saying thank you very much to Sydney and Inside Scientific for hosting this and sponsors with the American Physiological Society and Aurora Scientific. So I'm delighted to be able to take this time today to talk to you about a new technique that we've developed in my lab and studying and applying it to studying of myosin binding protein C in striated muscles. So with that, I will go ahead and get started. Let's see. Okay, moving right along. Okay, so to give you an idea of where I'd like to go today and what I'd like to talk to you about is for the uninitiated, I wanted to give you a little bit of an idea of myosin binding protein C and its family members, why we need a new approach to studying myosin binding protein C, or why we could benefit from a new approach, and then talk a little bit more about just a couple of vignettes that I'll have for you about data, structural and functional, about the roles of myosin binding protein C that we've discovered in striated muscle, uncovered by the use of this cut and paste approach. So that we're all starting on the same page, I want to point out that striated muscles are cardiac and skeletal muscles that are known as striated because of the striations that can appear, I'm looking for the laser pointer, that can appear in histological sections of the muscle here and are easily seen in electron micrographs. And so these striations are actually indicative of the basic functional units of muscle, that is sarcomeres, that run from these narrow black lines here in electron microscopy from one Z disc to Z disc. And then the sarcomere itself is composed, as I'll show you, of thick and thin filaments in a highly ordered, almost rigid lattice sort of array. Okay, so cardiac myosin binding protein C fits into all of this in that it's a sarcomeric protein that's present in the sarcomeres in thick filaments. So again, for orientation, these are Z discs here. And then myosin binding protein C is often observed as a series of longitudinal lines, 9 lines that appear on each A band of the sarcomeres. So this is just shown a little bit easier in cartoon format, Z lines here, linked to thin filaments. Thick filaments are where the A band is, here, this dark region in the sarcomere. These are composed of thick filaments and myosin that ultimately reach out to actin and undergo ATP hydrolysis to generate force or filament sliding here. C protein I've localized to this Z zone or this C zone within each half of a sarcomere. So that'll come back up later when we talk about fluorescent localization, that you'll just see one band on either side of the sarcomere here. But C protein, cardiac myosin binding protein C in particular, is the family member that has been studied most. And this is in part because mutations in myosin binding protein C have been linked to hypertrophic cardiomyopathy. And they're the most common cause of hypertrophic cardiomyopathy, as a matter of fact. And myosin binding protein C itself then affects the rate at which myosin cross bridges interact with actin. And it's one of those proteins that's phosphorylated in response to fight or flight stimuli. So it's necessary, it's critical for normal function of the heart, as well as in disease states, C protein is affected. So Although cardiac myosin binding protein C has received the most attention, what we've found is that skeletal myosin binding protein C paralogs are also linked to skeletal muscle diseases. So this has just been in the last few years, it's been found out that C protein mutations in the skeletal muscle versions of C protein actually are responsible for things like distal arthrogryposes, which are distal limb contractures, as well as more recently, it's been found that C protein mutations can lead to muscle tremors, which is kind of an exciting new finding that we'll touch on again at the end. So there are three, as alluded to, there are three myosin binding protein C paralogs that are coded by three separate genes, but they all overall maintain a similar structure, with the exception that the cardiac has an additional domain at the N-terminus of it. But otherwise, they're repeating domains of Ig-like or fibronectin-like domains, folded domains. And then they all have what's referred to as an M domain or an M motif at the N-terminus that seems to be one of the critical regulatory regions of this molecule. So there's a slow skeletal, fast skeletal, and cardiac version. So it gets a little tricky though, and this is one reason why the skeletal versions have been difficult to study, is that it's been difficult to study difficult to study either fast or slow myosin binding protein C, because co-expression of the fast and slow isoforms or paralogs occurs to some extent in all skeletal muscles. So this is not really, this image from Schifano is not related to myosin binding protein C, but what it's meant to do is highlight the intermixture of, in a single muscle, how many different fiber types there are. And then if you consider each of those fiber types is going to be expressing a variable degree of fast and slow myosin binding protein C, it sort of highlights the difficulty that one would have in trying to parse out what the function of the relative function of the different isoforms are. You combine that with the additional realization that the slow skeletal isoform encoded by myosin binding protein C1 gene actually has a series of splice sites in it, so that there's a family of 14 different proteins that are made from the slow skeletal alone. So when you combine the splice variants along with the co-expression, then it's turns out to be an incredibly challenging feat to be able to look at what each of these isoforms do on their own. To further complicate matters, or another issue that the field has been dealing with, is that it's clear that the myosin binding protein C binds to multiple binding partners. So the 8, 9, and 10 domains of all three of the isoforms actually are responsible for embedding C protein in the thick filaments. So the C-terminus of C protein is responsible for embedding C protein at these very characteristic localizations within the C zone here. But on top of that, it was understood early on that the N-terminal domains also interact with myosin, be it the myosin heads, the myosin neck, myosin regulatory light chains. And then more recently, oh, I skipped the punchline actually, I'm sorry about that. But in addition to binding to the myosin and myosin heads for the N-terminus, according to my lab and many others, we've determined that there's actin binding activity of these N-terminal domains as well. And then more recently, and to bring you up to date, these middle domains, which haven't been explored practically at all in terms of function, were found to bind to myosin S1 and S2 as well. So this has created quite a puzzle for those in the myosin binding protein C field for really envisioning how this protein interacts in the sarcomere, especially when myosin and actin are on different filaments entirely. So there have been a number of models produced, and one of which is something that we drew in 2011 with this idea that the N-terminal domains of myosin binding protein C are free to interact with actin, and that the C-terminal domains interact with the thick filament. and that we might actually be creating C links within the sarcomere at some point here. And then according to this model, when we drew it, I also put the N-terminal domains in the middle domains in conjunction, or in close proximity rather, to the myosin heads to allow for this idea that that interaction might work. So the question that's been occupying the field right now is, does C protein dynamically shift between these filaments, or are there distinct populations of myosin binding protein C? The issue here in answering that question, as simple as it might be, is the dynamic part of it, as well as the idea that we're up until now lacking some fundamental ways to manipulate myosin binding protein C within the sarcomere. So for instance, most studies or many studies that have been done about myosin binding protein C have used in vitro assays such as ATPase binding or ATPase assays and binding assays that, for instance, show binding to either actin or myosin. And then some force assays too, where typically only parts of myosin binding protein C recombinant domains are used, and the localization of C protein to its specific portion in the sarcomere is lost. So the alternative to that is transgenic animal models, where you can actually, C protein gets incorporated exactly where it should be within the sarcomere, and its localization is preserved. But there's a great distance between the in vitro assays and the animal models in terms of time and money. In addition, as helpful as animal models have been, and as much as I use animal models, there's always compensatory effects that you wonder, if you've introduced a mutation, are you looking at compensatory effects to that mutation or primary effects? So because of this, my lab had taken a look at this, and really we're pining away for a way in which we could just physically go into the sarcomere and cut myosin binding protein C out and then replace it, so within minutes. And this came from kind of a historic knowledge that many of the myofilament proteins you could biochemically remove and replace. And that's still done for many of the thin filament proteins. So we investigated a little bit, and looking for our cut and paste method, what we found, the cut was relatively easy to envision, is that tobacco etch virus has a consensus sequence that is very rare in mammalian proteins. So rare that it's often used as a way to have a tag when you're doing a recombinant protein, and then you can cleave that protein away from the rest of the tags. So that was straightforward. But the paste was a little trickier until we came across molecular superglues that were designed by Mark Howarth while he was at Oxford. He's now at Cambridge University. But these are nano-engineered split protein pairs. And so what this is, for instance, is this is a protein holodomain that Mark and his group split into a catcher and a tag domain. And when these two catcher and tags come back together, they form an instantaneous isopeptide covalent bond here, which is essentially a molecular superglue. So spy catcher and spy tag, which are shown here and which I'll refer to in the upcoming session or upcoming slides, were derived from Streptococcus pyogenes. And so that's where the spy designation came from. But they form a spontaneous covalent bond when they're combined. So it's almost like magic. So this has been published already, but I'm going to go through it here because this is the basic premise of what I'm going to be showing you. Is that what we did was we took the TEV consensus sequence and the spy tag, and we inserted those sequences into the myosin binding protein C3 gene, so that when myocytes were harvested from hearts of these mice, then what we could do is add TEV protease to the bath, and have TEV protease then recognize that site within myosin binding protein C and cleave off the N-terminal domain here. And then if we make a recombinant protein that has the cognate spy catcher on it, as shown here, then we can reproduce a covalent bond in the spy catcher spy tag combination that's shown here. This is just fluorescence localization of the normal doublet stripes of myosin binding protein C. When you take a homozygous myocyte and treat it with TEV, you remove those fluorescent N-terminal domains of the protein, whereas in control wild type, there's no effect. And then when you add back C0, C7 with the spy catcher, you reacquire those fluorescent images, or the fluorescent localization. So what this says is that you can exchange with any desired combination of recombinant protein in only minutes for testing very specific hypotheses, excuse me, of myosin binding protein C function. So what I wanted to show you here, and this is a really exciting part and what has motivated this talk coming together, is that we've expanded the cut and paste approach to skeletal muscles now. So we've made an identical analogous version of the cardiac myosin binding protein C spicy mice and made spicy 1 mice that are the slow skeletal slow skeletal isoforms. And what we're able to do then is it's exactly analogous, is that we've inserted a TEV cut site and a spy tag into myosin binding protein C1 gene. For a second group of mice that affect the fast myosin binding protein C, what we did was we've taken another orthologous pair of split protein pair called Snoop tag and Snoop catcher. I did not name these. These are Mark Howarth's design, and I encourage you to go to the handout section and also his website. He's got a lot of really nifty combinations of this. But the idea here is that the Snoop catcher will not recognize the spy tag. It will only recognize Snoop tag. And so this provides a way that we could selectively manipulate either slow skeletal myosin binding protein C or fast myosin binding protein C. We've also created, and this is a delight to tell you, that we've created our new, what we are calling double agent mice, because they're not only spies and snoops, but now they're double agents because they have both capabilities, in that homozygous mice, for the double agent mice, have all of their fast skeletal exchanged with the TEV site and the Snoop tag, whereas all of the muscle in the slow fibers is replaced with the slow myosin binding protein C mouse here, as in SPICE. So what this enables us to do is actually remove C protein from an entire muscle, regardless of whether it's fast or slow. So you can think of it as a double knockout without the consequent compensatory changes. And so just to highlight what we're talking about is that this double agent mouse, the homozygous animals that are viable and appear overtly normal at this stage, would have C protein knocked out, or we would have the capability to knock out C protein from all of these fibers here. Okay, so for the last part of this, I would like to give you two quick vignettes on how we have used the cut and paste approach so far and what we've learned from it so far. And I'm looking forward to questions on the cut and paste at the end, so please be sure to put them in chat. Okay, so what have we learned? Well, we've focused primarily on two different methods to really understand what myosin binding protein C is doing. But the first one is looking at structure. So we've been, the spicy mice really have given us an unprecedented ability to look at structure in the same fibers over time, so with repeated measures. And so I'll show you that in a second. As well as we've now used, and this is my plug for my beloved Aurora equipment, but we've got a suite of Aurora equipment ranging from single cell apparatuses, so permeabilized single cell apparatuses, to the 1700A, the myofibril apparatus that Matthew told you a bit about, and a bunch of things in between. And it's been amazingly helpful to study the function. What do these muscles, what kind of forces do these muscles produce? So we'll go through each of these vignettes quickly, and let's get started. So for the structure, we've teamed up with Tom Irving and Waikong Ma at BioCat, and gone out there a couple of times already for looking at the structure of muscle using X-ray diffraction. And these experiments also were conducted in collaboration with Anthony Hessel, which we'll talk more about, but Anthony Hessel and a graduate student in my lab now have these results under review. So we're really pretty excited about this collaboration that's come together really nicely. But basically, to give you an idea how the system is used and the types of data we get out of this, we have a SNP-C2 psoas muscle that's permeabilized, detergent permeabilized and glycerin permeabilized, I should say, and that we're able to take a look at the X-ray diffraction patterns under different conditions when there's 2.4, 2.7, or 3 micron sarcomere length, under relaxin conditions, so no calcium. And under these control conditions, we assume that the fast skeletal myosin binding protein C is intact. Then we add the TEV protease and let that incubate for 20 minutes or so, and we repeat our measurements. So the second, the repeated measure would be in the absence of myosin binding protein C, at least those N-terminal domains. So this is to show you a little bit more and convince you more about the technique itself. That here shows a Western blot of fast skeletal myosin binding protein C. And then upon TEV treatment, what you see here is the antibody recognizes the N-terminal fragment. And so that's the C1, C7 fragment that has been cleaved, the first N-terminal part of the molecule. And we know that that's a soluble piece of the molecule because then when we subject the tissues to washing and just subject it to gentle wash and without TEV, we're able to remove that band. What's shown here is in the SNP-C2 mice, when we cut with TEV, the slow skeletal myosin binding protein C probed with a different antibody is not affected by the TEV cleavage. So this is selective for the fast skeletal. So what I want to show you here is that this is condensing a lot of the X-ray diffraction data. And believe me, we have analyzed practically every line that we're able to. But to just give you a little vignette right now of what sort of things we're finding and what the results are, is that we looked at spacings that are associated with the myosin thick filament here. So the M3 reflection and an M6 reflection. And what I want to kind of show you is that we have the pre-values in blue, and the pre-values, for instance, for the M3 are known to change with sarcomere length here. So we have kind of an internal quality control for our data by showing that we're able to get expected changes in these bands with sarcomere length as expected. But what we find is, at least for the thick filament spacings, that when we post-TEV treat, that we've actually see an expansion of the M3 spacing and the M6 spacing, consistent with that C protein might have been restricting the thick filament in some way. There's many ways to interpret this data, so that's just one. And at this point, we're keeping other possibilities in mind, as you would see in our discussion section with this. What really surprised me, though, the most was that looking at thin filament signatures and reflections, what we found was that we also saw significant changes. But in this case, for instance, post-TEV, our G-actin spacing decreased, as if that there was some tension on the thin filament exerted by myosin binding protein C. So again, that's one interpretation, and we feel pretty confident in that based on other movement of troponin, that we've got a thick or a thin filament effect here. But what was the most surprising to me about this was that this implies that, or provides structural evidence in support of C-links in these relaxed muscles, that the changes in the thin filament that I just showed you, the changes in the G-actin spacing, are consistent with some sort of interaction of the N-terminus of myosin binding protein C. So we went on to look to see whether or not we could kind of corroborate any of these with any functional force measurements. And while I have a lot of force measurements, again, from all these different pieces of equipment, what I really want to show you is a phenomenon that I'm pretty sure that you guys don't see on a regular basis. And that's what you're looking at here is just a trace record, a force trace record. So this is arbitrary at this point. This is a KTR maneuver and a stretch activation maneuver. But what I want you to focus on is just how nice and solid this line is and unwavering. Whereas when we treat this we treat cardiomyocytes or even myofibrils with TEV, what we see then is kind of this wiggly or oscillatory, as I'll show you, behavior. And you can see it in the raw force trace. And then when you add C protein back, that behavior dissipates again. So this is just showing you another trace where under control conditions, the force trace is really nice. Post TEV, you see these oscillatory traces. And then when you add the C1, C7 recombinant back, you can dissipate them. So what that actually looks like in real time. Oh, you've got to be kidding me. It's got to work. Hang on. There we go. Okay. So it's a wave-like motion that continues throughout the tissue and is attributed, and so these kind of gross disturbances in the force are attributed to that. We had actually published this already in cardiac and noticed that this is how fiber looks when it's in calcium activating solution before the treatment. You see the oscillations, and then after addition of the recombinant protein, the oscillations dissipate. So what these are have been described for quite some time, starting in actually the 1950s. But they're a period of sarcomere rapid lengthening. I'll get my pointer back again. Rapid lengthening here, followed by slow shortening or contraction. So Fabiato and Fabiato gave a really nice description of this in 1978. And they pointed out, and this is the important part, is that SPOC originates from sarcomeres and occurs in the absence of sarcoplasmic reticulum. So these spontaneous oscillatory behavior is a sarcomeric emergent property, is one of the messages that I want to get across to you. And thinking about this, I've thought about SPOC as relaxation uncoupled from a decrease in calcium. So for instance, this is the typical increase in force of a twitch that you would happen to see in a cardiomyocyte that lasts as long as the calcium is applied. But in SPOC, if you lose myosin binding protein C, or if you phosphorylate it, which I haven't shown you, you're predisposed to these this SPOC or oscillatory conditions, where to me, the difference is that we have premature relaxation. Then when you return C protein to the system, then you again restore the calcium coupled activity. So now this goes to the small area that I'll talk about with that kind of meshes up with the first part of Matt's talk, and it's to look at myofibril activation and relaxation. So I have to say, as a full disclaimer, I got interested in Matt and Aurora's system precisely to do these measurements to test whether or not SPOC could be related to changes in relaxation. So this is a graph, or this is a typical trace that is developed, and I should have this marked as where the red starts, where calcium activates a myofibril, and then calcium is taken away so that we can measure rates of activation and rates of relaxation. Thing that I really wanted to talk about just briefly is that if you were to zero in on this very tip right here and expand the time course, then you get what's referred to as a slow phase of relaxation, where this slow phase and the time it takes in this very slow but short linear phase is due to cross-bridge detachment, whereas the rest of this relaxation phase here is thought to be due to more of a catastrophic giving between sarcomeres or within a sarcomere, and probably due more to structural effects rather than cross-bridge effects. So taking a look at losing, going back to cardiac myosin binding protein C and cleaving off the C terminus, C0 through C7 on myofibril kinetics, what we see is that actually there's not much change in the total force that myocytes can exert, and there's not much change in that K active, or the K activation rather. So it's the beginning part when force is increasing. And we didn't see much effect on the slow K-rel in this experiment, and I qualify that in a second. But where we did see dramatic effects for myosin binding protein C, the loss of myosin binding protein C in the pink bars here in the TEV treated, is that we saw an acceleration of that fast K-rel. And then we also saw a propensity for SPOC to occur. So it didn't occur in all of our myofibrils, but it occurred in a substantial proportion of them, and more than in wild type, or the wild type reconstituted, or the wild type starting fibers. So the take home from that is loss of C protein primarily affects that kind of structural effect of the sarcomere. And so we're interpreting this in terms of that C protein slows relaxation through two mechanisms. And the first is that this is functional evidence for that cross-linking that goes along with some other data that I haven't shown you here, but I'm happy to talk about, where we believe that that fast K-rel is due to changes, and that is due to this cross-linking here, as well as data I haven't shown you do point to this idea that C protein can activate the thin filament and slow the slow K-rel, or the cross-bridge detachment rate here. So to bring this kind of full circle again, then one of the things that has motivated our studying of C protein over time is really trying to understand what this protein does under normal circumstances, but then what goes wrong that can cause diseases. So in that sense, I was really intrigued by a few articles that have come out from colleagues' labs, and in particular from Katya's lab. There have been really amazing work that she's done on skeletal isoforms and mutations of myosin binding protein C. And really wonderful review article here that actually begins to point to sarcomeric proteins, and mutations in them is associated with tremors. So this is kind of a new paradigm to think that there could be that mutations in sarcomeres have myogenic causes that then can lead to muscle tremors. And so this is a departure from a neuro kind of centric basis for central, or for muscle tremors. And putting this all together with what I've shown you is I'm keenly interested in determining whether SPOC is one of the factors, SPOC and impaired relaxation, that could be one of these myogenic causes that affect tremor here. So what we're doing now is introducing mutations into C protein using the cut and paste approach to investigate mutations both in cardiac and skeletal muscles, and look at whether or not they have effects on this sort of tremor-like behavior. So what I've told you today, or I hope convinced you of, is that C protein is a complex family of proteins encoded by 3 separate genes. It gets very complicated, though, to sort out their function because they're alternatively spliced, and they have many post-translational modifications that alter their function. So because of this, in the plethora of proteins there were, we came up with a cut and paste approach for a new methodological tool to study the effects of C protein in working sarcomeres and preserve their location in situ. And then what I've hoped to show you is just a piece of how we're using the cut and paste approach, sorry about that, to revealing new structural and functional roles of C protein within the sarcomere. And watch this space to be determined if spot contributes to skeletal muscle tremors or other muscle arrhythmias. So that's the direction that we're heading with this, and defining more of the molecular mechanisms by which C protein affects sarcomere function. So with that, I'd like to thank everyone who has contributed to this work, and in particular, all the members of my lab who have been working hard on the spicy mice and breeding them at this point. And then special thanks to Anthony Hessel, who has taken us under his wing for teaching us more about X-ray diffraction, as well as Tom Irving and Wei-Kong Ma, and Devin, who's a graduate student in Tom's lab. So with that, I really thank you for your attention, and I'm happy to take any questions.
Great presentation, Samantha. Thank you so much for that. I'm going to bring Matt on now to join you. Okay, so let's just jump right into our Q&A session now. The first question, can the cut and paste approach be used in living cells?
So I want to say yes, and that is actually a direction that we're excitedly moving. And I think everything is in place to back this up a little bit. I'm envisioning delivery of TV protease to living cells, and that could be AAV injection to mouse hearts, for instance. And actually, I'm very encouraged because I know of at least 2 groups, and I don't think they've published, although they've presented this work, so I don't think I'm disclosing anything. At least 2 groups have succeeded in expressing TEV protease to cleave sarcomeric proteins that have a TEV site in them. So it's a matter of the technology is out there, and we're moving in that direction to do it as well.
Okay, really great. Thanks for the details. How efficient is replacement of the myosin binding protein C with recombinant domains?
So far, when we've done it with the spicy stuff, or the spy catcher, spy tag, that's 90 to 100% efficient. So what's really limiting is making sure that we aren't diffusion limited, or that there's some other buffer condition. But the spy catcher, spy tag itself is really high fidelity in that basically as soon as they come together, then the reaction goes 100%.
Okay, fantastic. Thank you. Just a reminder that the Q&A tab is where we can collect all your questions, so feel free to send those in for Dr. Harris or Matthew. Our next question, can the cut and paste approach be used in other proteins?
I'm hoping so. I actually don't know at this point. We haven't tried it with any other ones, but I'm really encouraged by how well all 3 mouse models that I have, or 4 of them now, are really cut well with the TEV protease. And then there's a titin cleavage mouse out there that was developed by Wolfgang Linke and is used by Anthony Hessel, for instance. And that works really, really well. So my guess is that this could be easily transferred to another protein, so long as you know regions that you can, so long as you know something about the protein going into it.
Okay, fantastic. Here we go. We have another question that just rolled in. Is it known whether the myosin binding protein C loss is associated with cardiac arrhythmias resembling Spock waves?
So thank you very much. That is like the question that I really want to know. I'm not sure that there's any evidence that you would just like whether or not C protein causes arrhythmias more than mutations in myosin heavy chain, for instance. So I'm not sure that falls out of the data yet, but I'm very, very interested in asking in answering that question whether protein loss is associated with the arrhythmias, because for HCM mutations with C protein, most of those result in less protein produced. And so I would predict that we would be able to induce SPOC more easily. And under conditions, say, of calcium load, that's exactly the hypothesis. I would expect to see that we would see more arrhythmias in myocytes or in hearts that had less myosin binding protein C. And so we're testing that, as a matter of fact.
That's very exciting. Great. Thank you. Do you both have a couple of minutes if we go over just to answer the next couple of questions?
Yes, I'm actually really happy to.
Great.
So the next question, I swear I didn't plant these, but I love these questions, you guys, so thank you. Do you observe SPOC in traditional C protein knockout models? The answer is wholeheartedly yes. And so therein is actually something, how I knew about SPOC in the first place is that I was lucky enough to work in Rick Moss's lab on the first C protein knockout mouse that was produced, and I would see SPOC all the time. And I would see SPOC in wild type myocytes that were phosphorylated as well. And, I asked Rick about it at the time. We kind of puzzled on it. But at the time, and this is really, really key, SPOC has always been viewed as kind of a phenomenological thing that people see maybe when their myocytes aren't feeling so great, aren't the best quality. And because if you saw the knockout hearts for C protein, they're really awful, mangled looking hearts that we just kind of shrugged it off. But it always stuck in my head because I went and I did the steps to investigate what SPOC was, find out who was really modeling it and what was understood about it. And so it's been always something that I've been aware of. And so when I saw it as a phenotype in all of our spicy and snoopsy mice, then it's become really clear that this is a phenomena that's related to regulation by C protein.
Oh, great. I'm glad that you like all these questions that popped in.
So yeah, I'll try to be quicker about it, but
that's okay.
Okay, what sort of effects of C
Yeah.
protein mutations on contractile kinetics, filament structure, etc., have you seen, if any? So yes, actually, it about killed me to pare down my talk to just these few slides because that's exactly where we're going with it. We've got a ton of data showing that mutations, especially in the cardiac, because we've worked with it a little bit more, have an effect similar to some transgenic mouse models we have, and we can replicate it using the spicy system, for instance. So it seems like it's a high fidelity system that really replicates a lot of the basic changes in kinetics that we see. And so right now, kind of as a foreshadowing to biophysics and all that, we're really looking at the middle domains as well and really diving into that because besides doing some binding assays, there's not been a lot functionally done with them. Let's see. So aging is associated with serious muscle mass and strength loss. Any changes in myosin binding protein C. So that, again, I couldn't have, paid you guys off to do this better, but yeah, that's something that we're actually looking at right now. We've become interested in aging for a number of reasons, and I think there's one report out there, and I really, really regret that I don't remember the first author or the last author, but I think that there's one paper out there that does seem to indicate that C protein decreases in heart over time, and that is like a real question that we want to dig into. And right now I've got a couple undergrads playing around with that in the lab. So stay tuned.
That's great. I think we have time for one more question. And
so I think Matt has a question.
Yeah, I see that in there.
Yes, from my dear friend Alexi. Thank you for the question. Actually, you'll be very pleased to know that just this morning before the talk, our programmer and mutual friend, showed me a preview of the new algorithm that he's trying to implement. So I think it would actually be a big leap forward in terms of the one that's sort of in test now, if you will. So I can't say exactly when it'll drop, but it'll certainly be available for trial not long from now.
And Matt, for anyone who can't see the question, can you just
Oh, I'm
clarify?
sorry. Yeah, great talk. I know you are planning a new release of the HVSL, the high-speed video length sarcomere system with a new edge detection algorithm. When to expect it. So
perfect.
There you have it.
Insider knowledge.
That's it. That's why we have these things. Stay till the very end, folks.
Yes.
So related to that, I told Matt that I wouldn't get too gushy or anything, but I have to say, for anyone who's thinking about the MyFibril setup, I went with Aurora because they had it commercially available, and I've got years of experience where I can tell you personally that Matt and Chris will get on the phone with you practically any time of the day or night, especially if you're trying to get data right before an abstract is due, and we'll walk you through anything that you need for technical support of this sophisticated equipment. And it's been really a factor that's been meaningful for me in my lab.
It's most kind of you, Sam, to say that.
Yes, that's very nice. Okay, we have gone a few minutes over, so we will have to close it out here. If anybody has any more questions, though, you can submit it in that survey. So I want to say thank you so much to Matt and Sam for your knowledge and your presentations today. It was such a pleasure to be working with you.
Thanks so much, Sydney.
Thank you,
Sam.
Thanks, Sydney. Thanks, Matt.
In closing, we hope you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society and sponsored by Aurora Scientific, and I hope you enjoy the rest of your day.